Today we're going to talk about the urinary system. Now this system exists in our bodies to regulate the volume and the composition of our blood. It's important for removing cellular waste products such as urea and other toxic substances in the blood. It also helps to promote red blood cell production and has an impact in regulating water loss from the body. So as I've just said, the topic for today is the urinary system, and as I mentioned, a key role of this system is to remove waste products from the bloodstream. These waste products include things such as urea, which is a waste of protein breakdown. Uric acid, that's a waste product of nucleic acid breakdown, and these are all products produced by cells that must be removed or they become toxic. The urine that's produced that contains this waste material has to be stored, and we have the urinary bladder, which is an expandable muscular stack that can hold up to 1 litre of this waste urine. And then the urine has to be removed from the body, and that's what we call excretion. Now the volume and pressure of the blood is also influenced by the way the urinary system holds back or releases water or solutes from the blood. Finally, the urinary system has a role in promoting the production of red blood cells. In this slide we can see the major organs of the urinary system. We'll start off with the kidneys, which are located here. This is where the filtration and purification of blood occurs. The kidneys are paired organs on the superior aspect of the posterior wall of the abdominal cavity. They're bean-shaped, about 10 to 12 centimeters in length and covered with fat. They extend between thoracic vertebra number 12 to about lumbar vertebra number 3, with the right kidney slightly lower than the left kidney due to the proximity of the liver here. The kidneys are retroperitoneal, which means that they sit behind the peritoneum membrane network. Notice other structures of the urinary system. We have the ureters, which take the urine from the kidneys. We have the urinary bladder down here, where the urine is stored, and then the urethra, through which the urine is passed to the external environment. This slide here just summarises some of the key facts that I mentioned previously. Start off with the kidneys. The kidneys are the paired organs which sit on the posterior abdominal wall, and these are the organs responsible for filtering and purifying the blood. They're retroperitoneal and surrounded by fat, what we call perinephric fat. I mentioned also that the right kidney is slightly inferior to the left kidney due to the presence of the liver superior to the kidneys. If you look at the kidneys, you'll see a structure, an indentation called a hilum, and this provides access for structures such as the blood vessels and the ureter. We've seen hilum structures in other organs that we've looked at, such as the spleen and the lymph nodes. I also forgot to mention in the previous slide that there are glands, what we call adrenal glands, that lie on the medial and superior aspect of each kidney, and these are important endocrine organs. That is, they make hormones. The adrenal glands will be considered in a separate lecture. In this slide we have a transverse section of the abdomen to show the kidneys and the surrounding structures. Notice the presence of the kidneys here and the presence of the liver here. Now, because of the presence of the liver here, the right kidney is slightly inferior to the left kidney. Notice this fat, the perinephric fat that surrounds each of the kidneys. Notice also the major blood vessels. We have the abdominal aorta and inferior vena cava here. It's important to remember that a key role of the kidneys is to filter the blood. Notice also this white line, that is representing part of the peritoneal membrane network. The kidneys sit behind this, therefore they are retroperitoneal. This shows the basic internal anatomy of the kidneys, and you will see a model in the practical that looks fairly similar to this. There are two basic regions within the kidney. We have an outer cortex, about a millimetre thick, and a deeper inner medulla. And many organs that we look at in this unit will have an outer cortex and an inner medulla. If you notice here, there are extensions of the cortex, the tissue that we have in the cortex, there are extensions of this into the medulla, and these are called renal columns. These divide the medulla up and allow the formation of these medullary pyramids. If you look closely, you might be able to see how the renal columns provide access points for blood vessels entering and exiting the kidney. So if we now take a look at the medulla of the kidney, we can see the presence of these triangular structures, what we refer to as medullary pyramids, and we can see each one of these pyramids tapers towards a point, which we call a renal papilla. This is nestled into a cup-like structure that's called a calyx. A minor calyx initially, and then you'll get several minor calyxes which fuse together to form a major calyx. These converge upon the renal pelvis here, which is then continuous with the ureter. Now these structures here that I've just mentioned, the calyces, the pelvis and the ureter, are the structures through which the waste urine will be drained. Notice also the presence of the hilum here, where the ureter and the blood vessels enter the kidney. This just summarises some of the key structures that I've mentioned, and also some of the structures that you'll be expected to identify in the practicals. I mentioned how the kidney has a cortex and medulla. I also mentioned how extensions of the cortex project into the medulla to form the renal columns, and we could see blood vessels in those. The renal columns divided the medulla up to allow the formation of renal pyramids. These are the darker cone-shaped structures. They have a pointy triangular papillae that faces internally and is nestled into one of the calyces, and the urine will drain through the calyces and then eventually out of the ureter. In this section, I'm going to focus on the major functional components of the kidneys. These are what are called uriniferous tubules. These are very important for the ability of the kidneys to filter and purify materials from the blood. Uriniferous tubules consist of structures called a nephron and a collecting duct. And here we can see the nephron here, and we'll focus a lot of attention on this structure here. We have about 1 million of these per kidney. A key role of the nephron is to filter blood and help with the formation of urine, and this occurs by three main processes. Firstly, filtering the blood to remove wastes and other small molecules, recovering or reabsorbing materials from the filtrate that the body can actually still use, and extracting or secreting materials from the capillary vessels surrounding the nephrons, for this then to be removed in the urine. The nephron is what allows the kidney to regulate the blood volume and the composition. This slide shows a lobe of a kidney, a section of a kidney including both the cortex and the medulla, and it highlights where the uriniferous tubules are evident, as we can see here. Nephron and associated collecting duct. Notice the nephron. Key role of that is to filter the blood. It receives blood from vessels we call afferent arterioles, and they're indicated here. The blood will actually be filtered and then processed into urine within the nephron and the collecting duct, and the urine will be drained through the collecting duct, projected towards then the renal papilla, and it will then be collected in one of the calyces. Now just notice some of the structures that have been highlighted on the nephron. There's a component of the nephron here called the proximal convoluted tubule. We'll talk more about that soon. That's located within the cortex of the kidney. You also have a distal convoluted tubule component as well, once again located in the cortex of the kidney. This structure down here, which projects deeply into the medulla, is referred to as the loop of Henle, and the collecting duct here will end up projecting down through the medulla towards the renal papilla. Note also how we have a nephron over here, where the loop of Henle projects quite deeply into the medulla, much more so than the one on the right. This is termed a juxtamedullary nephron, and this type of nephron composes about 20 to 30% of the nephrons in the human kidney. These nephrons are particularly important for the development of salt gradients in the renal medulla, which are important for the ability of these structures to concentrate urine. In this section of the lecture, we're going to consider the components of the Uriniferous tubules, and in particular we'll focus a lot of our attention on the nephron. The nephron is the main filtration and purification structure in the kidney. Each kidney has about 1 million of these structures. A nephron consists of several distinct components. Firstly, over here we have the renal corpuscle. It's the main filtration unit for the blood. So here, blood enters the structure and is then filtered. The filtrate from the blood is then passed through the renal tubule here. The renal tubule is composed of several major components: proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Eventually, this structure will connect to a collecting duct and the urine will be removed, taken down to the renal papilla and collected in one of those calyces and eventually removed from the kidney via the ureter. Now the nephron plus the associated connecting duct is collectively referred to as the Uriniferous tubule. In this slide here, we're trying to highlight the functional aspects of the Uriniferous tubules, show you some of the major events that are occurring in each section. So notice here the interface of the Uriniferous tubule with the blood vessels. Really important to understand this because a key role after all of this structure is to filter and purify the blood. So what happens here is we have a branch of the local blood vessel supply. We've got a little artery here, and then we have this structure called an efferent arteriole that enters the renal corpuscle. This clump-like structure here is a network of capillaries we call the glomerulus, and what happens here is material from the blood in the glomerulus is filtered and collected in the tubules of the nephron. So here filtration of the blood is occurring, and in particular materials of a small molecular weight are being removed from the blood, and we'll talk more about how that happens in a few minutes. So first of all, we filter materials from the blood, particularly small molecules. This filtrate is then passed through the tubule and it's further processed. What will occur is materials the body may wish to reabsorb, useful nutrients, salts and water are recovered from the filtrate and returned to the local blood supply. In addition, toxic elements or waste products in the blood are passed into the tubule for addition to the urine that's forming there. So 3 key things are occurring. The blood is being filtered, useful materials are being then reabsorbed from that filtrate and put back into the bloodstream, and additional waste products and potentially toxic elements are also being added to the tubule as the urine formation occurs. Filtration, reabsorption and tubular secretion, the 3 key roles of the Uriniferous tubules. In this slide we're seeking to build upon some of the concepts that I've just mentioned. The Uriniferous tubule components that I've mentioned previously are the nephron and the collecting duct. The nephron consists of 2 main parts, the renal corpuscle, and I tried to highlight that that is the structure where filtration of blood is occurring. Small molecular weight materials are being removed from the blood, and the waste products and toxic products that we want to remove typically are the smaller molecules, things like urea and uric acid. The material that is then filtered out is passed into the renal tubule, and this is the structure where the blood filtrate is processed into urine. And what is happening here is useful materials such as water are being recovered and additional waste products are being secreted from the local blood supply into this tubule. The renal tubule consists of 3 key components: proximal convoluted tubule, loop of Henle and distal convoluted tubule, and we'll consider all of these as we're progressing through this lecture. The collecting duct is what is attached to the nephron, and material produced by the nephron will enter the collecting duct. Additional purification and waste processing will occur in the collecting duct, and it will then eventually take the urine down to the renal papilla in the medullary pyramid. It will then be drained into one of the calyces and eventually it'll leave via the ureter. Now a key fact to note is the Uriniferous tubules are tightly integrated with blood vessels. That's logical because a key role of the kidney is to filter and purify the blood. The roles of these local blood vessels are to deliver the blood to the renal corpuscle where the filtration occurs, and then also to facilitate further exchange of gases and nutrients during the processes of reabsorption and tubular secretion. This slide here shows the blood vessels associated with the nephron in particular. What we can see here firstly is the afferent arteriole. This is the vessel that is responsible for delivering the blood to the nephron. It enters the renal corpuscle, and in this structure here, filtration of the material in the bloods will occur. The vessel exiting the renal corpuscle is the efferent arteriole. This then forms a capillary bed that we call the peritubular capillaries. These are low pressure porous vessels, and they're found in the cortex, and they're specialized for absorbing materials from the tubule cells. So if we have material that's been filtered out, what we may wish to recover, materials such as nutrient molecules, water and some salts, the peritubular capillaries allow us to reabsorb that material. The vasa recta vessels we see down here, these are capillaries which extend into the medulla and closely parallel the loop of Henle. These vessels play a key role in concentrating urine. What I'll do now is spend some time talking about the specific sections of the nephron. We'll start off with the renal corpuscle. The renal corpuscle is the portion where the filtration of the blood occurs. This structure consists of two major components. Initially we have a cluster of blood vessels here, that's the red lines there, cluster of capillaries. These are inside the renal corpuscle. These vessels originate from the afferent arteriole, which brings the blood into the renal corpuscle. Now these capillaries have many pores or holes. We call these fenestrations, and these allow for the filtering of material from the blood. The other structure that we have in the renal corpuscle is the glomerula, or it's also called the Bowman's capsule. Now an important concept is that it is a two-layered membrane, so two layers of tightly associated epithelial cells lining the glomerulus. We have an inner visceral layer that's in direct contact with the capillaries. This layer then folds back on itself and forms an outer parietal layer. This is the layer that forms the outer wall of the renal corpuscle. In this slide we can see the relationship of the glomerulus to the glomerular capsule. Here we have the afferent arteriole delivering blood to the renal corpuscle. From this branches the glomerulus, the tuft of capillaries. They're fenestrated, which means they have small holes in them. The glomerulus is then covered by the double-layered glomerular capsule. Now one layer of the capsule is termed the visceral layer of the glomerular capsule. That is the layer that's in direct contact with the capillaries of the glomerulus. The cells that are part of this capsule are called podocytes. This capsule layer then folds back on itself and becomes the outer parietal layer, the parietal layer of the glomerular capsule indicated here. Notice the space between the parietal layer and the visceral layer. It is into this space that fluid filtered out of the blood will accumulate. Over here we have a microscopic image of the glomerulus and the associated visceral layer of the glomerular capsule. Look closely and you can see red blood cells. The capsule layer folds back on itself, and then this layer here, it's very difficult to see, but you might just be able to see the thin side of the cells. Between the parietal layer and the visceral layer there is a space, and this is the space where material filtered from the blood will accumulate. That filtrate will then be passed into the proximal convoluted tubule, and the urine processing will continue. What we'll now consider is how filtration of the blood occurs in the renal corpuscle. Inside the renal corpuscle we have set up a filtration membrane through which the blood is filtered. This membrane consists of three main layers. Firstly, we have cells of the capillaries of the glomerulus. These are termed fenestrated capillaries because they have holes in them, and the holes allow materials that are small in size, small in molecular weight, to exit the blood. There's then a basement membrane, as there is with any epithelial layer. There is then the layer of cells that we refer to as the visceral layer of the glomerular capsule that sits on top of these fenestrated capillaries. They're in direct contact with one another. The cells of this layer of the glomerular capsule are called podocytes, and they're characterized by slit-like extensions. And these two cell layers, those of the capillaries and those of the visceral layer of the glomerular capsule, establish a filtration membrane, and we'll look at that soon. This is showing you a model of the structures I've just mentioned. So this is a renal corpuscle here. It's a very detailed model. Notice the following structures: the afferent arteriole that delivers blood to the renal corpuscle, the tuft of capillaries that we call the glomerulus. If we look on this side, if you look very closely, you'll notice little holes or pores represented on these capillaries. These are the fenestrations. The capillaries have a layer of cells covering them, and on this side, that layer of cells has been left in place. They've been removed from this side. These yellow-looking cells are cells of the visceral layer of the glomerular capsule, and they are termed podocytes. And if you look closely, you can see they have this zigzaggy link between them. They form these filtration slits. And what happens is blood enters this structure, and then small materials in the blood under pressure are forced out through the holes in the capillaries and the slits between the podocytes. The material then accumulates in this space here. On this area here that I'm indicating now, that is the parietal layer of the glomerular capsule, and that stops the filtrate going out to the surrounding tissue. That layer here is continuous with the visceral layer. The other structure to notice here is the proximal convoluted tubule. Urine will be directed down here for further processing. This slide here emphasizes the structures that help form the filtration membrane. So here we have the glomerulus inside the renal corpuscle with the capsule on top of it. We'll magnify that and have a look at some of the key structures. Notice the capillary here. It has fenestrations, or little holes. They're about 70 to 90 nanometers. Small holes through which small molecules, such as waste products, can be removed from the blood. Sitting on top of the capillaries, we have cells of the visceral layer of the glomerular capsule, cells we call podocytes, and they form these interdigitations between each other, and these are then able to form the filtration slits, which help in the filtration of material from the blood. So the filtration slits, formed by the podocytes, the holes in the capillary, together they allow the removal of small materials from the blood, which include the waste products you actually want to get rid of. This image is to try and help you visualize the process of filtration which is occurring in the glomerulus. A key concept to remember is that blood entering this structure is under pressure, and this pressure forces small molecular weight materials out of the blood through the filtration membrane. So blood enters here under pressure, material is then forced through the filtration membrane. The system kind of works like this strainer of tea. Imagine the fluid and the tannins in the tea are like the small molecular materials in the blood. They can pass through the holes in the sieve and go straight through. Larger materials such as the tea leaves are retained by the strainer. An analogy of that in here is the blood cells and the large molecules. They are too large to pass through this filtration membrane. They will keep flowing through the blood. Only small molecular weight materials can be filtered through the filtration membrane, and this includes the waste products we want to get rid of. Once the filtration of the blood has occurred, the next thing we need to do is process this filtrate into urine. The blood filtrate, while it does contain waste products, it also contains materials that the body could use, nutrients, water and salts, so we need to recover that. It is in the renal tubule that's attached to the renal corpuscle where this recovery process occurs and the concentration of urine occurs. the renal tubule, as we see here, continuous with the renal corpuscle and composed of three main segments. Proximal convoluted tubule. Think about the naming of this. It's proximal to the renal corpuscle. Convoluted just means that it's very windy. There's then this hairpin shaped structure here, the loop of Henle. This projects deep into the medulla of the kidney. We then have the distal convoluted tubule, distal from the point of attachment of this structure to the renal corpuscle, convoluted because it's windy. Nephrons then attach to a collecting duct, which we can see here. So we'll consider these components in the next few slides. First of all, we'll consider the proximal convoluted tubule, and that's just highlighted briefly here. Renal corpuscle there, proximal convoluted tubule attached directly to the renal corpuscle. So filtrate from the renal corpuscle enters this structure first. The tubule is lined by simple cuboidal epithelium, and under the microscope what's evident on these cells are microvilli. Now microvilli we've seen when we looked at the digestive system. Microvilli increase the surface area and help with the process of reabsorption, and in this structure a lot of materials that you want to recover are being reabsorbed. Things like glucose and other nutrients and salts are being reabsorbed. Also some water. After passing through the proximal convoluted tubule, the filtrate from the blood enters the loop of Henle. It's a long hairpin-like structure, and the hairpin is a reference to this 180-degree turn that the tube makes. It projects deep into the medulla. It has ascending and descending regions, so as it projects downwards, descending, and then as it comes back up, ascending. It's also lined by epithelium, a single layer of epithelial cells. In some areas it's squamous, very flat. In some areas more cuboidal in appearance. In this area here, the loop of Henle will facilitate the reabsorption of a lot of water. If we look closely at the medulla, we can see a salt gradient, and that salt gradient is very important for encouraging the movement of water from the loop of Henle back into the bloodstream. So the loop of Henle, very important for the recovery of water, and also salts are recovered in this region here, salts that are potentially useful for the body. The distal convoluted tubule is the final part of the nephron. As indicated here, this is the portion just before we attach to the collecting duct. Now in this section there's also recovery of water and sodium, as we've seen in other areas. However, what can also occur here is that the recovery of water and sodium can be fine-tuned by the release of specific hormones into the bloodstream. There's a hormone called aldosterone, released by the adrenal glands, that will fine-tune the reabsorption of sodium from the filtrate. Antidiuretic hormone, released by the pituitary gland, it will control the reabsorption of water from this section. So if you're becoming dehydrated, ADH will trigger the reabsorption of more water from this particular region of the nephron. So in the distal convoluted tubule, fine-tuning of the processes of sodium and water will occur, and that helps you to match what your body requires at that particular time. So if you're dehydrated, you need more water is recovered as you process the urine here. In this section of the lecture, we'll move away from the kidneys and we'll start considering some of the other important structures of the urinary system. First of these are the urinary tracts, in particular the ureters. The ureters are continuous with the kidneys at the renal pelvis here. One can see there the hilum, that's where the renal pelvis will be found. And the ureters transport urine away from the kidneys inferiorly towards the urinary bladder, and that is where the urine will be stored. We'll also talk about the urinary bladders. We'll look at the basic structure of that. And then we'll look at the urethra, which is the ducting system, the tubing system, which removes urine from the body. Note also the local blood vessels here, abdominal aorta for delivering blood to the kidneys, and the blood is taken from the abdominal aorta through the renal arteries, left and right. The arteries and the urinary tracts entering each kidney at the hilum, this indented region here. Notice also in this diagram the adrenal glands are indicated by my arrow here, and that adrenal gland there and that one there, they are the ones responsible for releasing the aldosterone, which I spoke about in the previous lecture when I was discussing the function of the distal convoluted tubule. So here we have a cadaver photograph showing actual ureters. So here we have the kidney, adrenal gland, and there at the hilum, posterior to these blood vessels, we have the ureter associated with the right kidney. Now, the ureter can be considered as having a couple of different sections. There's the abdominal section that goes for about 12 centimeters within the abdominal cavity, and then once it enters the pelvic cavity, we refer to it as the pelvic part of the ureter. So important for taking urine from the kidneys down here to the urinary bladder, where it's stored before being excreted from the body. Local structures to notice: you've got your vena cava here, you've got your abdominal aorta here, branching into the common iliac arteries. Once the urine is taken down the ureters, it is placed in a structure we call the urinary bladder, and this is an expandable muscular reserve for your urine. It sits behind the pubic symphysis. That's that patch of fibrocartilage that links together the anterior aspects of the coxal bones. The urinary bladder sits posterior to the peritoneum, so it's retroperitoneal, although its superior aspect is covered by the peritoneum. If you look inside a urinary bladder when it's empty, you will see folds, ruga. We've seen ruga before when we were looking at the stomach. So when the stomach is empty, the internal surface, you can see these folds. These folds allow for expansion when food enters the structure. Same sort of thing is happening here. When empty, the urinary bladder has lots of folds, ruga, which facilitate expansion of the urinary bladder as urine enters it. You can have up to a litre of urine stored in the urinary bladder. As it fills, the urine distends the bladder superiorly until it assumes an oval shape. A structure that we'll point out in the next slide is called the trigon. It's an inferior posterior triangular area where we can see the points of entry of the following tubes: the ureters and the single urethra. Before I progress any further, it's important to mention that the ureters and the urinary bladders are lined with muscle. This is important. For example, the ureters, they are not just passive tubes. The urine does not go down from the kidneys to the urinary bladder through the ureters by simple gravity. The tubes are lined by muscle, which help to force and massage the urine downwards, not unlike the process of peristalsis we saw when we were looking at the digestive system. The urinary bladder also has muscle lining its walls, which is very important for allowing the urine to be excreted from the body. So this shows the basic anatomy of the urinary bladder. Remember, its key role is urine storage. When full, the bladder expands into the abdominal cavity and the walls get thinner. Notice we have ruga, which are evident, these folds on the inner surface of the urinary bladder. They allow for expansion of this organ. The trigon that I mentioned before, this triangular structure at the base, the posterior inferior aspect of the bladder, is indicated here. And what we have here and here are the openings for the left and right ureters. Here we have the opening for the urethra through which the urine will pass, and there are muscular sphincters in different locations to regulate the flow of urine through this bladder. Take a look at the wall of the bladder here, and it just highlights a couple of important points. Transitional epithelium is lining the inner surface of the bladder. This is a specialized type of epithelium that prevents the toxic elements from the urine going further into the tissue, so it provides a tight seal. This transitional epithelium also allows for expansion of the bladder, so it's able to accommodate the stretching of this organ that occurs as the urine is filling the structure. Other important layer here is the muscularis, muscles, layers of smooth muscle here that are important for allowing you to expel urine from the urinary bladder through the urethra during a process we call micturition. Finally, notice the peritoneum on the superior aspect of the urinary bladder. This organ is retroperitoneal, so it doesn't sit, it's not suspended within the peritoneal network, it sits behind it, it's retroperitoneal. This slide is simply highlighting the shape changes the bladder undergoes as it fills with urine. The empty bladder here appears collapsed, orange. Notice the relatively thick wall on this structure. When full, the bladder expands into the abdominal cavity. The walls get thinner, and there we can see that there. Notice the urethra. This is the final part of the urinary system we'll consider. This allows urine to be removed from the body. And the urethra is what we'll consider in the final slides of this lecture. The urethra is a thin-walled muscular tube leading from the urinary bladder to the body exterior. So just remember that concept I was talking about before. The ureter, the urethra, and the urinary bladder are actually lined with muscle. So the flow of urine from the kidneys is not simply passively occurring through gravity. There are muscle layers within the ureter, urethra, and the urinary bladder helping to regulate the flow of urine through the body. There are major differences between the female and male urethras. The female urethra is relatively short, 3 to 4 centimeters long, and tracks tightly on the anterior vaginal wall. The male urethra is longer and has 3 distinct regions. Part of the male urethra runs through the prostate gland. That's a gland that makes components of semen. Membranous urethra crosses a layer of skeletal muscle. Penile urethra is the longest one, and that runs through the body of the penis. This shows the basic structure of the female urethra. Notice the urinary bladder here and the relatively short urethra, which is tracking to the anterior wall of the vagina. And lastly, we have the male urethra. Here we have the urinary bladder, and here we have 3 distinct sections. Firstly, the prostatic urethra. This structure here is the prostate gland, which makes components that are present in semen. Then there's a short section where the urethra passes through a layer of skeletal muscle, the membranous urethra. Then we can see the majority of the male urethra is this section here that passes through the penis, the penile urethra. We'll talk about the erectile tissue structures that are indicated here. That's these blue coloured materials here. They're important during the erection process. So 3 sections of the male urethra, through the prostate, through the skeletal muscle here, and the majority of it through the erectile tissue that we see there. Prostatic urethra, membranous urethra, penile urethra. And that concludes this lecture.